Good afternoon and good evening, everybody. Thank you very much for joining us on this wonderful webinar, which looks at the use of monoclonal antibodies, or what we call biologics. We will give a bit of time for colleagues to join, but we are pretty happy that you're here to learn a lot from our excellent speakers and that you've joined us. As I said, we're just going to give it another 1 minute or two for colleagues to join us. While we're doing that, for those of you who have joined us already, perhaps you could just introduce yourself in the chat. How the session will work, of course, I'll give more detail. We will not have questions and answers until the end. But we will have a function where you can start actually populating your questions on the Zoom. There's a function there that you will be able to do that. So let's give it another minute and then we will basically start with the webinar. Thank you so much again for joining us and we appreciate it. We will try to keep on time, which is an hour and a half. Thank you. Good morning, good afternoon, good evening, everybody. Thank you very much again for joining us. My name is Lynette Mabote, and I am the current outgoing board member representing NGOs on the Unitate board. We are very excited to have this session today. This is our very first learning and development session as the NGO delegation, as well as the communities sitting on the Unitate board for 2024. This L&D session is very significant and timely, actually, as this is an issue that Unitate is exploring. We are hosting this webinar in collaboration and partnership with the O'Neill Institute for National and Global Health Law, the International AIDS Vaccines Initiatives, which is ZIAVI, Access IBSA, and Dondora Community AIDS Support Association, DACASA, based in Kenya. So looking at this very important topic because we are facing a crisis that needs to be addressed. Why are we having this session? I mean, we know that therapeutic antibodies, including monoclonal antibodies, are the cornerstone of treatment, treating and actually preventing diseases in most high-income countries. Yet, when we look at the African market, for instance, we only account for around 1% of the global access to monoclonal antibodies. These antibodies can say you have saved many lives and have many uses that can be used, including, for instance, saving children from respiratory syncytial virus, and looking at COVID. Some MABs are used during COVID, and looking at how people living with non-communicable diseases such as cancer could actually benefit from these. But we know that we face many issues, including inequity, and this is what we're going to be discussing today. We definitely want to see MABs being widely available in LMICs as well as in high-income countries. We want these to make sure that we've got equitable access, that the high prices, that the complex manufacturing processes, intellectual property barriers, as well as the regulatory challenges are overcome. So it is up to us as national, subnational, regional NGOs and communities to start forming coalitions around MABs in order to work in this area and to ensure that we've got a transnational access system of access to monoclonal antibodies. We've got a wonderful panel of experts who are going to be discussing many issues, some of which include the role of antibodies in the immune system, the benefits of the therapeutics of therapeutic antibodies. We'll be looking at the price as key challenges, strategies to reduce the cost of mAbs and how you manufacture them. We'll be looking at some of the intellectual property barriers to mAbs, regulatory pathway barriers, and then how we can actually come around and form an advocacy framework, because we definitely need to be moving up on this. We've got a wonderful lineup of speakers who all have expertise in the area who will be speaking. I'm just going to be calling them up. They will be introducing themselves, but we're very happy to have Dr. Matua, who's a medical director at IAVI. We've got Dr. Carmen Perez Casas, who is the senior strategic manager and PPPR head at Unitaid. We've got Sharon Ann Lynch, who is the co-director for the Center of Global Health, Policy and Politics at the O'Neill Institute for National and Global Law, as well as Patricia Osero Ochieng, who's the team leader at Dakasa in Kenya. Before we start, some quick housekeeping rules is that we must know that the Q&A function can either wait, or you can post your questions in the chat, or wait till the end of that presentation and then, of course, ask your question to a certain speaker. Because we're running out of time, and we will, of course, because it's such a dynamic topic, you will have an opportunity to actually speak to the speakers in the chat if you've got more questions after a presentation. So allow for that. Do know that the session is being recorded, and that the session will be shared, including the slides after captions have been added, will be shared on the O'Neill Facebook page as well as on the website. We will make sure that it's also shared widely with other parties who wish to, who have missed the session and want to know what happened. And just to let you know that once we have done with our five key speakers, we will have a short discussion session, and then we will allow you to ask questions and, of course, to forge a way forward as to how do we come up with an agenda, as well, an advocacy agenda around mAbs, and how do we situate it such that we actually have a lot of political will, we've got the funding, and that we've got the treatment literacy and the structures to ensure that we move mAbs forward. Okay, without further ado, I think we will now start. Our first presenter is, as I said, Dr. Matua. Dr. Matua is a medical director at the International AIDS Vaccine Initiative, IAVI. I hand over to you, Dr. Matua.
Thank you so much, Lynette, for that introduction. So as Lynette mentioned, I am working for IAVI, and I'm actually based out of Nairobi. So IAVI is involved in a lot of product development, including vaccines and antibodies. So I have about 10 or so minutes to just bring everybody up to speed with what antibodies are. So we don't want to assume that everyone understands what antibodies are. And then, just give you a very brief overview of, what antibodies can do, so that we can really open up the discussions. So IAVI is sponsored by multiple donors, and we just always take the opportunity to acknowledge the support that they give us. And the overview of my presentation, I will provide an introduction of antibodies, and then talk just very briefly about their therapeutic use, focusing just on cancer and HIV. But this is very brief because I believe other speakers will go into more detail of these therapeutic antibodies. So let's start with just the basics. What do we find in blood? So if you look at your blood, it has the red blood cells, which, as you all know, carries oxygen and facilitates exchange of oxygen and carbon dioxide between the tissues, the heart and the lungs. And then you have these cells called platelets. These are helpful in blood clotting. So when you get an injury, these are the ones that facilitate clotting. Just a minute, let me get my pointer. And then finally, we have your white cells, and there are multiple white cells with their own unique functions. So let's look at the different white cells and what they do. Again, if you remember your biology, we used to have two different types of white cells. We have granulocytes, and they're named because they have granules. I mean, it's kind of intuitive. So different granules determine how the white cells are named, and each of them has a unique function. For instance, the neutrophils will be more important for bacterial infection. Eosinophils are more common for parasitic infection and even allergies. Basophils for a lot of allergic reactions. And then we have a group called the granulocytes, which are the lymphocytes and the monocytes. The monocytes also fight off bacteria, viruses, and fungi. And the lymphocytes, then you have these three different types of lymphocytes that have various function. For production of antibodies, we really are talking about B lymphocytes. B lymphocytes. So every time you hear about antibodies, these are produced by a specific type of white cell called the B lymphocytes. And this really just gives you an idea that, the immune system is fairly complex. And so antibodies is just one of the ways that your body fights off infections. So let's now drill down into what antibodies really are. These are natural proteins, and from a structural perspective, we say that they are Y-shaped, as you can see here. So we have the stem of the Y, and this part of the antibody is very important in terms of determining what that antibody does. And then we have the arms of the Y. So this is Y-shaped, and the arms, the terminal ends of the arm, this is the section that is able to recognize anything that is abnormal in your body, be it a bacteria, be it a virus, be it a fungus, or even an abnormal cell. So the antibody is very specific. So for instance, if you're exposed to a bacteria, you can have multiple antibodies targeting different parts of that bacteria. They fit in, each antibody fits into that part almost like a jigsaw puzzle. So you get what we call an antibody antigen reaction, and that is what triggers the immune response. So as I said, antibodies are proteins that are produced by B lymphocytes, which are part of the immune system. Now, when we look at the antibodies, there are also different types of antibodies. So for instance,
Sorry,
you will have had...
Doctor, I think we actually needed like a whole 2 days with you. Sorry, you've got 5 more minutes to walk us through this. It's very interesting. I'm so sorry. Thank you. Continue.
No problem. So we have binding antibodies. These are able to tag bacteria. So they're basically, they alert the rest of the immune system that there is something abnormal and they need to come and deal with that. We have neutralizing antibodies. These can attach outside the bacteria and actually block them from causing the infection. And then the focus of today's discussion, we have monoclonal antibodies. These are antibodies that are produced by a single lineage of the B cell. So they're selected based on a particular and unique function. So if I can go very quickly over some of the monoclonal antibodies that we are working with. For instance, for HIV, we have been able to identify antibodies that can cross-react multiple lineages of the virus. As you can see, the virus is quite diverse. And we've been able to identify some antibodies that we call broadly neutralizing antibodies. We've been able to determine that these antibodies can potentially protect against HIV, and we're doing studies against that, can protect infants from getting infected, and can offer long-term protection. If I quickly go through the cancer treatment monoclonal antibodies, as I said, antibodies are able to recognize abnormal cells from normal cells because abnormal cells have different characteristics, and you'll be getting these slides, which you can look through. And so what we find from the antibodies is they're able to tag these abnormal cells and recruit other aspects of the immune system to destroy them. They can block these cells from multiplying, and then they can also be used to deliver drugs specifically to the cancer cells, and therefore minimizing the side effects. And you'll be able to see, this is my last slide. This is an example of a monoclonal antibody that has been used in breast cancer treatment. It targets a specific molecule on the breast cancer cells and prevents that cell from multiplying and prevents that cell from recruiting blood vessels that would otherwise increase its nutrients. So that's in a nutshell what antibodies do and what we can potentially use them for from a therapeutic perspective. Thank you so much.
Thank you so much, Doctor. That was very insightful. Of course, as I said, for many of us, we are going to need a whole 2 months with you just to get to understand, go back to biology, especially, and then, of course, to understand where we're coming from. But thank you very much for that wonderful presentation to start us off and to actually explain to us what it is that we're looking at. We're going to now move a bit forward, and we're going to be calling our next speaker, who is going to be speaking about the work that has been done. I mean, as I said, the reason why we are hosting this L&D session is because Unitate is looking at this area of work, and Unitate has been investing quite a lot of work around monoclonal antibodies, and this work is led by Dr. Kakatas. So, Carmen, let me just hand over to you for 10 minutes to walk us through the work that is being done at current point at Unitate and what is within this Unitate strategies for access to monoclonal antibodies. Over to you.
Thank you very much, Lynette. And it was a pleasure to remind indeed our biology lessons with you. Looking forward to check the slides. So I want to introduce today indeed what we are planning in Unitate, and very much welcoming and thanking this opportunity to share this with you. So first of all, we wanted to start by the problem statement, but obviously this is very well already introduced by you, Lynette, and by the prior speakers. We know that actually monoclonal antibodies are revolutionizing the pharmacopoeia for many different diseases. As we just saw, for example, in cancer or immunotherapies, they are actually increasing survival rates for some types of cancers very much, and that is happening in high-income countries. We also know that there is a great promise as well to be useful in low- and middle-income countries, and not only in non-communicable diseases, but also with a promise pipeline that is increasing for infectious diseases as well. We've been looking at this with many of you since years, especially, first of all, prone by HIV, when we were looking at the long-acting interventions, but then finding that monoclonals for HIV, which are also long-acting themselves, can actually be a powerful tool, not only in HIV, but in many other aspects. And finally, during the pandemic, specifically in COVID, and now in general for pandemic preparedness, antibodies are also coming as an important tool, because they can be one of the quickest countermeasures that can be developed and brought to the market very early. We will see later that, of course, there were many issues regarding availability of the COVID antibodies for low- and middle-income countries. So that was not materialized the potential in addition to the variants. But as I said, there have been recent breakthroughs in the infectious disease pipeline, and many others are in the near horizon for the infectious diseases, as you can see here, that matters very much as well in low- and middle-income countries. And so we are all looking at how to engage, how to ensure access as soon as possible, because the pipeline is not so distant. First of all, one of the breakthroughs is respiratory syncytial virus, optimized antibody, which appears here on the screen, that was approved very recently and have only in very few months in Northern European region, for example, in Northern Hemisphere, in the season of respiratory syncytial virus, we have seen a huge impact in decreasing hospitalizations by more than 90% in the countries that have been able to deliver this product this winter season to the infants and newborns. So decreasing hospitalization rates due to pneumonia and bronchiolitis has been amazing already. But we are also looking at others in the pipeline for that indication, and further down the line, a promise in malaria, specifically as well for babies, but for other use cases that could be very, very important in conjunction with the vaccine to eventually decrease incidence of malaria. And the same for HIV, and we will hear later about that as well. but the role of broadly neutralizing antibodies in prevention of infants transmission during breastfeeding, but also, of course, of other uses of the HIV antibodies for treatment or prevention. So we are looking at it with a lot of excitement, but we do know that access must be dealt with before and now, if we actually want to see that promise being materialized, not only in the North, but also in low and middle income countries. So you already said, Lynette, that actually 1%, only 1% of the total market now for antibodies is in the African region. They represent that a small part of the cake. So this is because of many different access barriers. I will summarize very quickly, but first of all, in innovation and availability, we have seen a complete, a complete lack of products that are adapted, fit for purpose for use in low and middle income countries. So there is no sufficient innovation in adapting the formulations, which remain cold chain, infusion, or intravenous use there. Also, the pipeline of infectious diseases is slow, but as I said, is somehow now being increasing with some products of interest. In the second barrier, in affordability, of course, prices are huge. And I know as well, Sharon will be coming into that. So just to comment that, for example, monthly treatments for some of the indications that IAVI saw are over $10,000 per month. And when there are generics, few of them, they are actually also unaffordable still. Even one shot, for example, for the respiratory syncytial virus monoclonal antibody I described is equivalent to $400. So we do need absolutely to work on price, but it's very difficult because of the manufacturing complexities, the IP and trade barriers that Achan will describe later, and which are very much difficult compared to small molecules, the traditional medicines we are using for those diseases. In addition, we know there is no sufficient capacity to produce. We saw that in COVID-19, and also it's not adequately distributed. It's very concentrated. Finally, the same for regulatory processes. They are really very complex, and there is lack of capacity in the authorities in the South because of lack of experience with the biologics. And I already mentioned the concentration of manufacturing, and in terms of demand and adoption. Honestly, there is so little experience that is a lot yet to be discovered there on how these products can actually be used, specifically in a complex armamentarium, if I can use this word, because we have to use them together with many other things, as you know, like maybe with vaccines, maybe with other preventative measures, for example, for some of the diseases. How this is all going to work around is a big issue. So we did a consultation where many of you were present, and that was last year with IAVI and Medicines Patent Pool and Wellcome and Gates to actually look at this very attentively and having expertise from all angles, diseases, manufacturing, IP, etc., and found out that there were maybe potential ways to actually address this. The report with the recommendations is available, and also a manuscript is going to be published. But just one of the recommendations I'm going to describe today very, very briefly is to actually work in a portfolio approach. So each of the monoclonal antibodies that is targeting a different disease will require different market interventions, because some of them, as I'm trying to represent here, might be only addressing very niche medical problems. For example, Ebola. We have heard that for refilling the stockpile, for example, of the World Health Organization as a reserve for potential outbreaks of Ebola, we might need only like one batch of production, 200,000 units per year. So that is not sufficient itself to be viable for a manufacturing company. There are other products that are addressing malaria or dengue, which are very big volume diseases in terms of burden. Majority of patients in low and middle income countries, but also interesting for other sides of the world. And finally, there are those like respiratory syncytial virus, I mentioned, the pneumonia for kids and bronchiolitis or HIV that share a market in both. So what we expect to is being able to actually now at this stage plan a consolidated approach, a portfolio approach that can ensure by availability of these products and access. So moving forward then on what we can do in Unitate. So as you know, access is in our DNA. So of course, knowing that access is the main barriers now to think on using these products when they come out of the pipeline, we have presented an area for intervention to our board that was approved, and subsequently we launched a call that I'm going to describe in the next slide. The area for intervention is focused actually in this part, anticipating the business models that could be viable to reduce the barriers for access for monoclonal antibodies, ensuring reduced cost of production, better products, and a simplified use as well. We try to do this in a product agnostic way so that we can actually be, having benefits for several of the diseases that worry today the world. We are also supporting other enabling elements, such as interventions in intellectual property through the different grants that we have and MPP, but also the regulatory processes as well. And we expect to work in the future in more concrete interventions for particular monoclonals that might need Unitate's added value as they move in the pipeline. That is, for example, supporting clinical trial capacity or supporting the implementation. And then as well, facilitating the adoption, because as I said, we know this is going to be quite difficult. We have learned a lot with long-acting, and we are learning many lessons could be applicable for this if they remain injectable, but there is a lot to do. So finally, just to say this will match with our strategic objectives as well as our programmatic priorities in our strategy from malaria to humans and children's health or HIV and co-infections, as well as the long-acting work in cross-cutting. So this is my last slide, is the call for proposal summary, but you can find it in the website. It's now closed, and we are now quite busy analyzing the proposals, hoping to share good news with the selected projects very soon. So that is all from my side. So thank you very much. Over to you, Lynette.
Thank you, Carmen. That was just wonderful. And thank you very much for situating the piece of work within Unitate's strategic plans, and also speaking about how it's being linked to a portfolio approach, which means you're not only looking at one product at a time, but we're seeing in terms of groups, how can we use this? And that is how Unitate saves lives faster. So we thank Carmen very much for that wonderful presentation. Those of you who've got questions around this very important area, please do ask us questions. Have a conversation with Carmen in the chat box there. She'll be able to just speak to you, because truly we are shifting the paradigm. As we shift this paradigm, we do know that we will be facing a lot of regulatory and IP barriers. Our next speaker, Achal Prabhala, who is the coordinator of Access IBSA, will be speaking about how we can try or work towards addressing the IP and regulatory barriers in order for us to move this agenda forward in a collective manner. Over to you, Achal.
Hi, I think actually that there's been a glitch in the program schedule, and I believe Sharon Ann is supposed to go on before me. So shall we, Sharon Ann, is that true? Is that?
Yes, I am the ghost in the machine. So yes. Sorry about that, Lynette. We gave you the wrong run of show.
Okay, no, that's fine. I mean, Sharon, please, you go, and then Achal will have you. So Sharon will be speaking to us about, the fact that as Carmen stated, we can do this. So there really is a pathway. Sharon will speak about, what does this pathway look like, and what are the opportunities out there that we can start looking at in order for us to increase the advocacy for this area of work. Over to you, Sharon. Thank you.
Thank you so much, and thank you for everyone that has joined. So I wanted to run through some of the information that the team at O'Neill has collected, and big thanks to Luis that has done the bulk of this. So first of all, let's look at the number of MAPs that are on the WHO Essential Medicines list. So there have been 11 that have been added since 2013. 5 have biosimilars versions at least registered in the US, in the EU, and a number in the US. I want to note, as Louise has done here, that with few exceptions, it is taking over a decade for MABs, once approved, to be available, to be listed as on the WHO Essential Medicines list. And I will be doing a bit of a deep dive on trastuzumab. And I just want to flag here that it was first approved by the US FDA in 1998. Part of the reason that MABs have been delayed, deferred, and outright rejected from the committee's review of the essential medicines list is because of price. This listing, as well as the comments, will be available in this, and more detailed comments will be available in the slide deck. So, as state of access of MABs, you've heard some speakers talk about this. It's important to acknowledge that in terms of the R&D pipeline, MABs represent the largest component within the biologics category of medicines that are undergoing research and development now. Also, it's the fastest growing sector financially in terms of revenue generated. Still, 80%, and thank you to IAVI and Welcome's fantastic report on this, 80% of sales have been concentrated in the US, Canada, and Europe. Looking at trastuzumab alone, there is essentially, outside of Western Europe, outside of Japan, outside of US, that market represents only 5% in terms of what has been bought and sold. So ROW is how Big Pharma, frankly, refers to the rest of the world. And this is where 80% of the population lives, as we know. Now, some of you that are online have been part of larger access to medicines and medical innovation efforts, and this might ring as a bad memory. In 1999, ARVs in Africa represented only 1.3% of the global market, that is, of ARVs that were bought and sold. The same is true now of monoclonal antibodies. So I guess if we were to have Prince come and back from the dead and re-record his song, we would be doing "We're Going to Fight" like it's 1999, because it is, and there are so many of the same barriers, the same excuses that were used not to provide the necessary political efforts, as Lynette has said, funding and innovation to close this gap. So just in terms of having it on one slide, the challenges and then the strategies to address, and this goes to one of the questions that was asked in the Q&A box. So we know that MABs are high priced and high over the reasonable cost of goods, incredibly high. So even though we're going to be talking about the fact that MABs are more expensive than the small molecule drugs, including ARVs, that are used in treatment of HIV today, that more can be done given that high markup. So obviously there is, similar to other lessons learned, of the competition, in this case from biosimilars. And we'll look at some of the examples that Louise found in terms of the downward pressure on innovator prices. In terms of the expensive and complex manufacturing, listen, we know that for, again, taking ARVs, because that 1999 is just rattling in my brain, the golden rule is that it takes 7 to 8 to 9 to 10 years from, let's say, US FDA approval to affordability, availability, and access in LMICs. And we have all been banging our head up against this, and to try to put the enablers in place to accelerate that time to population. Still, unless there is concerted action, that is going to look like a pipe dream when it comes to MABs, and frankly already has. Again, we'll be looking at trastuzumab as an example, and keep in mind it was approved by the US FDA in 1998. As Carmen mentioned, there is a lack of commercial incentive for big pharma to invest in health priorities that for LMICs, and also for them to invest in strategies and explore ways to lower the cost of goods, that is the cost of manufacture, or to even optimize. And we're going to talk about some of those strategies. And Achal will be speaking about the other challenge, which is the regulatory barriers at the WHO and national level, and what could be done about that. So to get to the very important issue of bending the cost curves, now MABs are essentially costing $100 a gram to manufacture. A goal, and it's a goal that experts say can be achievable, is a target of $10 a gram. Some of us have been talking about the need for a collective effort and indeed a consortium so that we can have ministries of health on board from the get-go. Civil society, of course, manufacturers, of course, and developers, global health actors, and donors, to take this framework, this target, and explore the various strategies to manufacture, reduce the cost of manufacturing. There are alternatives to manufacturing, such as different ways to make MABs. Right now, the Chinese hamster ovary cells is the mainstay, but there are various efforts taking place. We can't afford to have 1,000 flowers bloom and just hope that one of these will be the trick for the vast majority of MABs. Instead, we need to be organized. We need to have that innovation agenda, that optimization agenda, to fund and explore and adopt those strategies to lower the cost of MABs and manufacturing. As outside of manufacturing, there is still work that can be done. Similar to squeezing water out of rock, we can optimize doses in order to make doses more effective, lowering the dose, thereby requiring less API, thereby reducing the cost. Also, to be able to have MABs last longer in the body by elongating the half-life. Finally, we can look at better delivery mechanisms so that we don't have that barrier thrown in our face time and time again in terms of, acting as if all of Africa is a desert. It's the same arguments against that were used against ARVs in 1999, that MABs are too complex to administer. The reason we need this collective effort is, yes, to look at the bending the cost, but there's quite a lot of hesitancy out there and resistance because of the cost, because of that operational feasibility. So if we bring governments on board now, share the findings now, share what the experts say could be possible in terms of affordability, and make sure that people understand the safety and the efficacy, then we could have champions instead of opponents to this issue. Also, this would help to serve to soften the ground, obviously, and to give LMICs a greater role in the research and development pipeline, all with the principle of accelerating access and that time to population, and as well to adapt the innovations as they come, the innovations in optimization of the dosing and of reducing costs. The other strategy is, when I say leave MNCs behind in terms of the high costs and the lack of commercial incentive, I mean that there are strategies, and of course there are sources of manufacturing outside of those multinational corporations. We're going to be talking about some of the prices from Biominhos in Brazil. Cuba is manufacturing. There are other areas, obviously, in India as well. There has been a proposal, and I think a smart one, to create a cell line bank. And that would be where innovators work from a common pool and contribute to that pool of cell lines, thereby facilitating follow-on innovation as well as innovation to begin with. So back to trastuzumab. So trastuzumab is used for HER2 breast cancer, as I said. Here is essentially the price for what would be 18 cycles of treatments across various countries. You see a high of 68, it's nearly 30,000 for Kenya, 41,000 for Finland, etc. These prices are unnecessarily high, and keep in mind these are the originator prices. Just in looking at Kenya, and we'll hear more from Patricia at the end of the webinar, is that the cost of trastuzumab is still 9 times the average household income in Western Kenya, leading to incredibly low access. Now, again, that biosimilar strategy, the competition from biosimilars can have an impact. And for trastuzumab alone, the first time that there was a biosimilar introduced, it came in 15% lower than the originator. By the time of the 5th biosimilar, it was 58% lower than the originator brand. In addition to offering a viable, more affordable option, like with ARVs, we find that there's downward pressure on even the originator pricing. But still, just look at this comparison, where in Biominios, the price is 65% in Brazil, that of the originator brand. In Slovenia, and Luis has provided a couple of examples here, you see the stark difference in price between the brand Herceptin, the originator price, and these biosimilars as well, roughly less than a third, but it reduced the originator price to come down to closer to the biosimilar price over time. Another example, this is for Humira, one of the fastest, the highest grossing MAB that has been sold. And in Slovenia, again, look at eventually where the originator price dovetailed to match that. Now, some of the MABs you've already heard about, and I'm sorry we didn't. Thank you. I'm sorry we didn't get to hear more from Ayavi in terms of how do the MABs for malaria work, for HIV work. These MABs are in development and malaria is farther along. Given that this is the first MAB, NIH has a product as well as some of the developers that is specific to endemic areas and LMICs and is 100% funded, the development of, from philanthropic and public sector prices. So you better believe that we demand more, that we won't accept business as usual. So it is an opportunity, a catalyst, not only to make a way for a malaria MAB that right now shows efficacy at 84% in terms of reducing acquisition of malaria disease for pregnant women and children under 5, those that typically get left behind or at the end of the queue, because MABs by their nature are more targeted and safer. We can now have the most vulnerable groups to disease such as malaria be at the top of the queue. Thank you very much, Lynette, and everybody else.
Thank you very much, Sharon. And I think that was actually beautiful in situating what we do have in the pipeline or what we could look at and where we could go. But of course, there's always the gatekeepers. And the gatekeepers, of course, will not keep the gates closed because we've got Achal. Achal, I was actually, I've been following you for a very long time because you've been doing IP work and other diseases, HIV, TB, malaria for a very long time. When Sharon was talking about 1999 and talking about deja vu again, all I thought about was time after time. But hey, Achal is here to talk to us about how we can try to start addressing the IP and regulatory barriers in order for us to have further access. Also, Achal, if you could just also go a step back and kind of like situate us around monoclonal antibodies and biosimilars. Is it the same thing? Is it not? Is it biosimilar, a generic? Just a tiny bit of treatment literacy for 2 minutes. And then, of course, you can get into your presentation around how we work around these other barriers. Over to you. Thank you.
Thank you very much, Lynette, for the very warm introduction. And now that it is my time to speak, I'm glad that I'm doing this after Sharon Ann, because I think what she provided was a lot of the context that will allow what I say to make sense. And I think that the best way to begin, perhaps, is going back to 1999 and looking at what happened with HIV and AIDS. And she's absolutely right in that we are very much in exactly a similar moment where it's clear that MABs have great value in treatment, which is why they're being used so extensively, only in certain parts of the world, only in rich countries primarily. But what I'm here to do is also situate us in how it's very different as well. Because the thing with HIV and AIDS, and especially in 1999, was that we were dealing with small molecules. Small molecules are, and I apologize because there are many of you here who would have heard this a million times, but hopefully some of you who haven't, and it's for you that I'm doing this. Small molecules, of course, are products of chemistry. They are simple formulations relatively, which means that in general, both their intellectual property protection as well as the pathways for regulatory approval are relatively easier than either of these two systems are when it comes to more complex drugs like biologics, which is a large category that technically includes vaccines. On the one hand, those are biologics which also contain, which reference immunology, as well as monoclonal antibodies, which are a very popular kind of biologic drug. And while I just very quickly set up my slides so that I can share my screen with you, I'd like to say that what's been really frustrating, I think, in the last 4 years especially, is that the dialogue that exists today around MABs appears to be very similar to a popular dialogue that existed at the beginning of COVID, which is especially around mRNA vaccines, which I found very frustrating, which was, oh, hey, here we have these 2 brilliant mRNA vaccines that are primarily being used in the United States and Europe. But obviously, for a range of different reasons, they cost too much, they're hard to store, they're extremely hard to make. No one in developing countries is really going to be able to use them. And it became a sort of self-fulfilling prophecy because it connects to a vast cycle of events that can be put into motion. If you wanted to make RNA vaccines available, there were many opportunities, most of which were closed on the basis of what I believe are false assumptions. I'm just going to set up my slideshow, which should be operational now. Can you see it?
Yes.
Great. All right, great. And so to start off with, so I'm going to look at 2 things. I'm going to look at intellectual property barriers when it comes to biologics in general. And then I'm going to look at the drug regulation system. So when it comes to intellectual property around biologic drugs, let's start with the problem. The first thing to know is that the patents that surround a single biologic drug tend to be really extensive. They can be in the hundreds, in the 200s even. They are also what's called an estate. All patent families are generally an estate, but the biologic patent estates tend to be very vast. And you can think of it as a huge piece of land where some part of that land is owned by one large farmer. Some bits of the land have been leased from others. Some bits of the land have more limited use rights. So it's sort of a conglomeration of various different parcels of land with different kinds of rights. And that's roughly what a biologics patent estate looks like. The problem with them is that there is nothing identifying them as being a part of that biologic drug that you're interested in. The owners are numerous. The categories even that the technology belongs to can be numerous. What makes it worse is that there are no specific classification codes for biologic drugs. that are adequate, either internationally or nationally. And what I mean by that is there are, in India, for instance, there are patent classification codes for biotechnology, which is an imprecise category. So some biologic drug patents fall under that category, many don't. It's not a useful category. And as a result of this as well, and this is a bit strange because we've had biologic drugs now for a very, very long time. Technically, vaccines are biologic drugs. So in fact, we've had biopharmaceuticals for hundreds of years, and biologic drugs in the way that we know them for many decades. And yet we do not have specific patent examination guidelines for these applications when they come in, as a result of which we found in a large research study that I led and in a paper that we published between the Access IPSA project and the Third World Network, I think one of whose co-authors, Chetali, is here, numerous other co-authors were involved in, that the patent examination, when it happens almost across the board, whatever country you're looking at, tends to be a little random. And so for the very same kind of situation, you'll have one patent application that's granted, and then on the other hand, another that is denied, right? But there isn't any governing logic to it, because for the governing logic to exist, the patent offices would need to have a manual as to how they examine biologic drugs, and they don't. It's also very hard to compulsory license these patents because the estates are large and unwieldy, and they're hard to identify. Now, this is a problem we ran into in India and South Africa, which have almost exactly similar situations where, for instance, the only way to create a product-based compulsory license is through government use. Now, government use is a way by which governments of countries can, in a situation of emergency or a crisis where they need to override patent protection, can do so relatively easily. And yet, government use has never actually been put into play, either in South Africa or India. So it's never been used even once. And we think that we can use government use to compulsory license all the patents on a product, such as, let's say, trastuzumab, because it does not ask for patents to be listed in the government use provisions in both these countries. For instance, however, compulsory licensing of any kind in both countries requires patents to be listed, which makes it almost an impossible situation to do so on a biologic, because unlike small molecules, you don't have a situation where you have one primary compound patent and you have maybe 6 or 7 other secondary patents attached. Brazil is a little more complicated, but it's roughly similar as well, in that there is ambiguity as to whether you can effectively have product-based compulsory licenses. So that's a problem. A couple of possible solutions. We could begin by developing classification codes for biologics. If we believe that they are an important therapeutic advance that we're living with today, that is not even a frontier technology, but something that we're very much surrounded by, then they deserve to have a classification code, either internationally or nationally, or both. And they certainly deserve to have rigorous and specific examination guidelines. There's an opportunity for international organizations to create generalized guidelines that countries can then translate into national law. But there's also, I think, an opportunity in countries where there is productive capacity to make biologics to go ahead and create their own guidelines that are then tailored to the principles that national patent law espouses, because those are different. And there is, I think, a very big opportunity to evaluate how and if you can have product-based compulsory licensing in countries, right? And even to the extent that it exists as a privilege that governments can exercise, it isn't adequate, especially in countries like India and South Africa, where governments have never exercised that responsibility. It needs to be available on a broader basis to larger sections of society. Compulsory licensing needs to allow for products to be compulsory licensed, as well as patent applications individually. Before we get to drug regulation, where this ties into, I think, both of these systems and the broader story that we're interested in telling is that while we all seem to agree that MAPS or biologics in general are an important therapeutic category that people need to have access to, the story of why they're so expensive and therefore then available and sometimes in a cyclical fashion is the dominant one as far as biologics are concerned. And so it's almost seen as sort of as a lost cause, as being unsuitable for countries. Whereas part of the effort that I'm trying to do here is to explain how the reason they're seen as unsuitable on the basis of price and availability, productive capacity, are largely a set of artificial reasons that we have under our control to change, but we do have to understand what they are exactly in order to do so. So the second monopoly barrier, in our opinion, around biologics is the drug regulation system. But here, there is a silver lining, which I'll get to in a second. But the problem is that primarily, if you look at the policies and laws for the approval of biosimilars or biologics in almost any country in the world, the drug regulatory system as it exists now is outdated. It's based upon a set of principles from at least 20 years ago. It places unreasonable conditions for biosimilar approval. And what that means is that the cost burden and the time burden for a company, facing a company, let's say, that wants to bring a biosimilar to market is unreasonable and is daunting. You have comparative clinical trials, which are a must. Let's say in India, for instance, right now, you have animal trials, which are a must, and these place an onerous burden, especially when you look at advances in medical science and in regulatory science, which have suggested that these are unnecessary. The regulatory burden in general, in terms of cost and time, translates to a much higher risk, which is a massive disincentive that prevents companies from readily, easily engaging in biosimilar market approval and development. These are small companies. You know, we think of generic pharmaceutical manufacturing in India as generally highly profitable. There are, of course, some very highly capitalized companies. But in general, even for these companies, the difference between, let's say, investing $20 million and 6 years in a process versus investing $5 million in 2 years, it's a significant one. These companies are not operating at the level of Pfizer. And this incentive has an easier way out, happily, which is in April 2022, the WHO revised its own guidelines for the regulatory approval of biologic drugs and released them. They are a substantial improvement on the status quo. In a country like India, for instance, they would reduce the time taken to develop and bring to market a biosimilar, as well as the cost, by up to 1/3 of what it is currently. However, India hasn't changed its regulatory pathway. We still have the same one. We still have the same code one. It still exists, even though it's been 2 years since the WHO issued new guidelines that so significantly change the development pathway. So we have this incredible opportunity to take the WHO guidance, which was hard fought for and hard won, and implement it into the policies and laws of developing countries that we're working in. And if we did so, we would be creating massive incentives for the productive capacity in those countries. Look, this might not be a step that everyone can take right now, and there might be more pressing things to be done, like the more basic infrastructural upgradation in order to produce biologics, because not every generic pharmaceutical manufacturer, especially if they've been concentrating on small molecules, can immediately produce biologics. But there are plenty of countries which already have that productive capacity across Latin America, Asia, and the African continent. And none of those countries have the best available laws that create the best available incentives based on the best available international guidance to bring those biosimilars to market. My colleagues, Chetali, Gopa, and I, and with several other co-authors, worked on a research project for about 3 years to examine in detail what the intellectual property barriers and what the regulatory system barriers look like for both biologics and for vaccines. You can get in touch with me if you'd like for more information, and the paper itself, which is fairly long and dense, but contains actually several pages that are devoted exclusively to analyzing the situation with monoclonal antibodies, is available at that address there. Thank you.
Thank you, Achal. You had a bit of time, but thank you very much for sparing us some time, because I can see the questions are blowing up. So you should also just get over there and start addressing some of those if you feel happy to, or we will actually have time to allow for a bit of a discussion. Our last presenter, but of course not the least, is a very powerful person who I think was one of the protagonists that led the HIV movement within Kenya, and is now again, deja vu, time after time, is back again and ready to fight when it comes to access to monoclonal antibodies. I would like to hand over to Patricia Asero Chieng of Dakasa to please take us through to what sort of advocacy framework should we be thinking about, and what sort of call to action to our governments must we start articulating as a group. Sharon Ann said, we need a movement. What does this movement look like? Patricia is one of those people who can help us start thinking about that. Patricia, over to you. She doesn't have a presentation. She will just be speaking. Just one last reminder that this is the webinar is recorded. The slides will be shared with you once the captions have been added, and we will have the recording up on the O'Neill website, as well as the Facebook page, as well as other sites. So it will be widely accessible for those of you who are asking. You will have the presentations. Over to you, Patricia.
Thank you so much, Lynette. You've given me a very big heart. Yeah, my name is Patricia Asero. I'm the chair of the International Community of Women Living with HIV, ICWK, the Kenya chapter. I'm also part of the community delegation of Unity. In Kenya, I sit in the Treatment Technical Working Group. And currently, I also sit in the subcommittee of advanced HIV disease. So I come with those different hats, but mostly the most important thing being here is that I'm a treatment advocate, and I believe that access to treatment and diagnostics is a human right. So based on those, I want to look back on what Dr. Mutua took us across. When we are talking about advocacy, first of all, as the presentations were being made, I was looking at the different speakers and picking out what is it that we can do. When Dr. Mutua came on board, I heard Lynette saying that we need 2 days. If Lynette, my teacher, needs 2 days, you can to understand when we are
Patricia, perhaps you could try. We can't hear you, so maybe you could switch off
You
your
went on mute.
it
But
muted.
now you're off mute.
Oh, okay.
No, you're
Yeah.
all good now.
I'm good. Where did I stop?
You were talking about what we need to do, learning, treatment.
Yes. So first is treatment literacy for communities, because we need our numbers and we are all over. So for us to be able to have our numbers, we have to be able to give treatment literacy so that our communities can be able to understand when we are talking about therapeutic antibodies, what are we saying? And why are they so important? Because so much of this is known in the West, but I can assure you that in Africa, this is a new thing for us. Most of us are not very much aware of what Sharon Ann speaks about very passionately, and she understands it very well. We are like, okay, and what is that? So treatment literacy, Lynette and our colleagues on this wall, that is first when we are talking about advocacy. But as we all know on this wall, we are all advocates, that our goal is access to MAPS. That is the goal of the advocacy that we intend to do. The second bit about that is about policy. because we will not have these maps in our countries without proper policies. So policies. I know that we have colleagues like Nitet who are lawyers. We'll have to bring in our lawyers, our think tanks, to be able to let us know what are those areas of policy that we need to be able to have in our different countries, and also at WHO. What are those policies that we are able to put in place that can move us forward? The third aspect was when Achan was talking about the regulations. When he spoke, I felt like, wow, this is not possible. Is it possible? And then as he went on, I had him talking about governmental use. And he brought me back to the year 2000, when Kenya was supposed to become TRIPS compliant. And we did not have medication. Some of us had lost relatives. We were working in facilities and people were dying. And we didn't know what to do with that. Then Kenya is supposed to be TRIPS compliant, and we are told, okay, the TRIPS has flexibilities. And one of those was governmental use. So we were able to push that in. But as Achana said, I've never had the Kenya government use governmental use. But he has brought it out so well that this is an area that we can be able to explore when we're talking about maps. So Achan and colleagues, I think this is an area that you need to educate us upon as advocates, that we really to get to understand these areas where we can get a leeway in terms of regulations, in terms of IP, so that we are able to understand what is that difference from the HIV drugs and what is this difference of the MABs, so that we can be able to internalize it and also explain it to our communities, but also be able to articulate it in any platform where we are, to be able to push to have access. I would want to see a place where I get these maps from the US, possibly Washington, to my village along the Lake Victoria. This is something I would want to have in my village. So what needs to happen for me to be able to make sure that the people in the village along the lake who are selling their fish are able to access that? And why do I talk about the people along the lake? Recently, you've all been seeing the floods in Kenya. Some people along the lake died, they were washed away, they don't have homes. And these are people who earn less than $1 a day, less than $1 a day. And some of them don't earn anything at all, maybe for a whole week, maybe $1 or $2 in a week. And as Saronan said, some of these medications are $400 US. So for a person who earns less than $1 a day, whether we speak of inequity, this person will never, ever have access if we don't do anything about it. And I believe as advocates, we have what it takes to be able to move this ball forward, because we have been able to identify the goal. We want access. And if we want access, we have within our ranks people with the different knowledge. If we will be collaborating, then the other component that we'll need to look at, who are our partners? Okay, we already have Unity, who is doing something. We have IAVI. I think IAVI is a partner. I don't know, Sharon will let us know. Are they a partner? Are they our allies? Are they people we can work with? Is Wellcome Trust a partner? Who can we rely on? WHO? Who are our partners? So as advocates, that is an area we need to find out. Who are our advocates? And because this work is so, there's so much to it, where are we going to get resources? Because we cannot do this work without resources. Where are we going to get resources? And then also need to find out who else is in this work, who is already doing something. Because as I was looking online, I found that IAVI and Wellcome already had a plan to action sometimes back for the same, to make sure that monoclonal antibodies-based products, the access is expanded. Is it possible for us to take up the call to action that is already out there, or do we want to come up with another call of action? Because I believe that as advocates, we have what it takes. If today we have more than 25 million people on antiretroviral medication, when in 2000 we were crying And then we stood up and said, enough is enough. And we've been doing our best. People are now on treatment. Now we are talking about non-communicable diseases. That is what our people are dying of. We are talking about advanced HIV disease. So we know that MABS is the way to go. If MABS can be able to help us stop all these things, then we have what it takes, advocates. You are out there. All it takes is for us to organize ourselves, because I believe we have what it takes. Where there's will, there's always a way. So back to you, Lynette.
Thank you so much, Patricia, and thank you so much for being bold as always. I mean, again, you are the one we take back to time after time, because you were there in 1999. Now we're back to 1999 with the MABs. And thank you very much for highlighting the fact that, we need to actually do our scoping properly, but I think we have done a lot of that groundwork. So, that is something that at least we have done. We're prepared of, we know the partners, we know definitely we need treatment literacy, we know we need the resources and who's doing what. It's how we then coordinate. Thank you very much for framing that for us, Patricia. I would just like to call on Chitali Rao from Third World Network to speak to some of the work that they've been doing around MABs. Chitali, if you could just come and just speak to us for the next, 3 to 5 minutes, just on the work that you've been doing, and then we'll get into a Q&A session. I am so sorry that we're running out of time, but we will have still 15 minutes of answering questions and of having a discussion. Chitali, over to you. Thank you.
Thank you so much, Lynette, Sharon, and Carmen, and Achal, for giving me the opportunity to speak here. I hope I'm audible. I'm currently at WHO. I hope, yeah, all right. So, yeah. So, regarding the work that Third World Network has been doing, actually, we were the WHO revised guidelines that were there was a long-drawn process of 8 years, which we had been following regularly with WHO, and it is very heartening to see that the guidelines have now been changed, and now I think there is a push to incorporate these guidelines into the countries, actually, because it is actually the countries' biosimilar guidelines that need to be changed for making these MAGs accessible. And there is also a very new welcome development that the EU has just come out with Europe. EMEA, the regulatory body in Europe, has come out with a new reflection paper, where they have said that they want to change the regulatory guidelines. As Achal pointed out, I must tell you that the three barriers that we have, the IP barriers, the regulatory barriers, as well as some of the barriers like lowering the production cost, as well as the pre-qualification of WHO. All these factors are cumulative factors that give rise to the high price that we are seeing with respect to MABs. So for one of these, as Achal pointed out, the regulatory barriers is also very important. And I am not talking from a perspective of reducing the cost, but I'm also talking from the perspective of science-based approach, because we are not cutting corners with respect to when we are asking for reducing the timelines with respect to both the animal clinical trials as well as reducing the human clinical trials, the efficacy trials, because both of these are science-based approach which has been followed globally, and there is scientific evidence to support this right now. So that is very important for people to understand, that it is not just from a cost perspective that we are compromising on the safety of the patients. This is from a very scientific advice that we are coming from. So I think it is time that all the countries do recognize the fact that MABs, monoclonal antibodies and biologics, are actually the need of the hour. If we talk about the cost of these drugs, as Sharon pointed out, this is humongous. And a lot of countries have the capabilities to produce these MABs, especially countries like India and Brazil. And it is important that we tap on these resources to make these products available. And the second point that I'd like to highlight is with respect to the IP barriers that Achal has pointed out. I think it is time for our IP officers also to look at new examination guidelines that can be brought about with respect to these, because that was pointed out in one of the papers. Getting 238 patents for only a single monoclonal antibody does not justify actually the cost of these drugs, because if I talk only from the perspective of the drug Humira, more than 80% of the patents were filed when the drug was already approved by the FDA, and it was in the last 10 years of 6 years of its expiration. So this actually talks about the marketing strategies that has been implemented by the companies to just keep the patent thickets alive and to just not reduce the cost of the drugs. There is a need for these drugs globally, and I think it is time that we also look at the new pharmaceutical guidelines that can be implemented with respect to this. So, and the last point that I will like to point out is with respect to, I think Sharon also pointed out, the pre-qualifications. There is a lot of companies globally that can produce these maps, but the problem is that these are not pre-qualified. So the cost of pre-qualification for these companies is very high. So I think we need to work with WHO to look at how these pre-qualification standards can be reduced, or how we can work around these, so that a lot of these products are capable of getting into the LMICs. So I think Sharonan has been advocating for this for a long time regarding the pre-qualification of the monoclonal antibodies. And I think we also need to look at WHO's pre-qualification programs to see how many companies can make it to that pre-qualification. That was, thank you so much.
Thank you very much, Chitali. Please do stay on the line because there may be questions for you. We're now opening it up. We do have some time. We've saved some time. We've got 13 minutes. If all the speakers could just also open up the cameras so that we can have this conversation. Some questions have been asked and have been answered, but I would like to actually allow 1 or 2 people to raise their hands and ask critical questions if they have. I think I'm just going to also just steal some of my discretion as the chair or as the moderator to ask the first question. And this is to, I think, to both to Carmen, to Achal, as well as to Chitali. And it's speaking to WHO's slowness in changing all these guidelines and how we can actually help colleagues such as Sharonan in promoting this and pushing for the pre-qualification, because I think if we don't get that moving, a lot of our countries will still take the 10 years to even want to shift the needle. That is the first question that I have. And number 2, the second question that I have is, why has it been so impossible for us to look at this area? Then why did we only start looking at it during COVID, and why our government's still using the same excuse as expense when they have seen what a pandemic like COVID can do? So that is to our speakers. And then, of course, I ask you to please ask your questions. Some of the speakers can take some of those questions that I've asked while we collect others. Thank you. Okay, must I pick on people?
Yes.
I like picking on people. Achal, I'll pick on you. You were right there.
I want to just say something mildly controversial, which is I think that to the extent that I found that in India, for instance, we didn't get any of the reformulated booster vaccines that people in Europe and America got. So 2 different reformulated booster vaccines for the mutated strains of COVID. And the entire country of India did not have access to them. There was also no demand for them. And this was also rather convenient for our government, because it meant that they didn't have to do something that people didn't feel was necessary, even though it was. And I think that some of this is also the situation with MABs. It's also the situation with biologic drugs, right? I think that we were speaking to Patricia's point, is I do think that this is a hard chicken and egg problem to solve, but I do believe that there has to be a better understanding of what we need, what we deserve, and also a better articulation of the fact that we need it. That then moves people like our government in India or others to then figure out how to fulfill that need, right? But at the moment, this is an unasked want. There's one tiny question that a point that Brooke made in the Q&A. I have a terrible keyboard and I just have a bad time typing, and it's easier to answer. Brooke is saying that government use typically would allow for product-based compulsory licensing, so it's covered. What I want to say is that that's a little complicated. It allows for product-based compulsory licensing by default because it doesn't demand that patents be listed in a government use application. I also will say that for me personally, it's wholly inadequate. Chetali and I and Gopa are working on advocacy in India. It's wholly inadequate because the Indian government has never used it. We believe it's also wholly inadequate in South Africa because the South African government has never put government use into play. And so the idea of having a potential option is not terribly exciting to us. And I think that it has to be extended to compulsory licensing as well, which definitely does not allow for product-based compulsory licensing. So that's the broad ask.
Thank you so much. And Chatali, would you like to add?
Lynette, I would like to answer your question on the comments on the slow guidelines for the WHO. So the thing is that it took 8 long years to move WHO to change these guidelines. So I think TWN has been working on it for the last 8 years, and finally in 2022 we saw the light of the day when actually, and it was then actually the scientific committee had twice rejected the regulatory guidelines to be changed. This is ironical because there is scientific evidence and WHO is a harbinger of guidelines globally. A lot of other developing countries, developed LMICs, everyone follows the WHO guidelines. So I think processes like these where we know that there is a lot of need of the hour for the medicines, I think this should not be such a long drawn process. So that is, TWN is now incessantly working with a lot of other developing countries also to change their guidelines. And we are taking the WHO WHO guidelines still has some loopholes in it, which we are trying to work on, but we are taking these guidelines, but you know, it is a slow process. And I think countries should also look at the science behind these guidelines. And I think then that will be an easy path for everyone to change when globally everyone is changing the guidelines. I think it should not be a problem to understand what is the basics behind it. So I think for later runs, it should be like, for example, I was also saying for vaccines, we also talk about an abbreviated pathway like Achal and we were discussing in one of the webinars. I think it should not be such a long drawn process for them also to bring these to, I think, to see the light of the day. So I hope that globally with the EU and the other Brazil and all of them looking for the new guidelines based on scientific evidence, UK has already changed their guidelines. So I think it should be relatively easier for us now to convince the countries to change their regulatory guidelines and make them more accessible.
Thank you very much for that. Before I hand over to Sharon Ann, I just see a wonderful comment from Louis da Gama, who is, the board member for the communities delegation, whom we're collaborating with, asking a question. Louis, would you like to add your bits, please? Would you like to take the floor for a minute or two, please? While Louis is preparing himself, I'll just allow Sharon Ann to come in. Sharon Ann?
I just want to draw some broad themes here since we're coming a bit to the close, and also to mention that in case you didn't see it in the chat, when it comes to feasibility, and Patricia challenged us, right? She was like, what about the people around the lake? And here she was talking about those that have lost homes, those that are near destitute because of crises there. I just want to flag that for some of the MABs in development and that are emerging, we hope, from the pipeline, for trastuzumab, the innovator has developed a subcutaneous injection as opposed to an IV. You know, even with COVID, we had people who should know better when we thought MABs could really make a difference with COVID, saying, well, it's not feasible. Tell me what isn't feasible about, and then it was an issue of hospital beds. You don't need a hospital bed to administer the COVID MABs. There are infusion centers that exist. We're not talking about deserts here, but still, it is so important, these innovations. So subcutaneous formulations for trastuzumab. Of course, the innovator took the opportunity to extend its monopoly, so that very much needs to be challenged. If that wasn't a call for a compulsory license, I don't know what is. And secondly, Carmen points out that the formulation for the malaria chemo prevention, the one from the NIH, the latest version, is also subcutaneous, right? So we are advancing on feasibility. Not all MABs will be able to be delivered in this form. And we're advancing on affordability. But you know, as people have said in the questions, how do we get there? How do we get the necessary funding, the political will, the proper innovation? And we can't look to the MNCs. We have to pretend almost that they don't flipping exist, because they don't care about these countries that are not attractive markets, right? And we see leadership, though, and possibilities coming from South Africa. Biominhos is developing MABs, and some of the MABs are developed by them for the same reason they started making ARVs. They provide a constitutional right to every person in Brazil, every citizen, to access to health care. And the MABs that have been needed, if they bought from the MNCs, it would have broken their bank and exploded their budget. Lynette has already flagged and complimented Unitaid, and I want to as well, for recognizing that addressing MABs can't be a single disease approach, that we need to be able to bring about changes in the way that MABs are conceived of, developed, and delivered across diseases. This is a platform issue. So thank you for that. And I also just want to ring the bell in terms of this one consideration. When there was a discussion recently among HIV researchers about MABs. And of course, I try to raise the issue, and they said, thank you very much, you treatment access activists. Let's get the product and then talk about access. You all know how flawed that is. But I also have to flag that research and development is itself being rationed because of the cost of MABs. So are there clinical trials going to be taking place for looking at adult HIV prevention and adolescent HIV prevention? No. Why? Because of the cost. WHO has postponed, deferred, denied MABs on the WHO essential medicines list, as you've heard, because of the cost. So it's a 1999 moment, and I'm so sorry for talking so long, but thank you.
No, that was wonderful. We have a question from Brooke. Brooke, if you could ask your question. And then we've got one from Peter. So let's get those two questions out. We are at the hour, but I don't know how the Zoom's just going to kill us, but we would appreciate like just a few more minutes so that we do hear these important questions. Brooke, please go ahead.
Well, I guess our challenge is to think what are the special areas where civil society has capacity to do things now that might make a difference, and where Unitaid, because of this call in particular, has special expertise. To me, I like the way Sharon Ann presented things, because it was the big picture of all the things that need to be done. And yet everything can't be done by particular individuals, or entities, or by civil society. We have to prioritize to some extent and stage our activities. And I think that's the harder question that we may not have been able to drill down in this session. So maybe we should consider again. To me, the product and process optimization are very, very key, because once you prove some feasibility, I agree with Sharon Ann that there's too much skepticism at present, but there still are really fundamental feasibility issues, and they weren't made up. You know, the reason things didn't happen because of COVID was fundamentally because of those feasibility issues, even though ACT Accelerator tried to some extent to bring monoclonal antibodies to bear. I urge Unitaid, as I always do, to not just talk about voluntary licenses, but to actually acknowledge the things that Achal was talking about, the possibilities of compulsory licenses, and to really clarify that in this area, given the complexity of the patent landscapes and the invisibility of the patent landscapes, a focus on compulsory licenses through government use, emergency means, or even competition-based licenses, or judicial licenses are all superior to at present in terms of TRIPS compliance. It really just kind of, sensitize this move for regional production to realize that governments are going to have to take some action to support this. They're going to have to support the research priorities. They're going to have to support the procurement, and they're going to have to support freedom to operate. And I know that, NGO delegation and some others are constantly accused of spending too much focus on intellectual property issues, but they really do end up being fundamental to whether there can be alternative producers. And right now, even though Sharon Ann says, well, let's ignore the big companies, they have some products that we will need. They will be in advance for a while, and we still are looking for more R&D from developing countries, and it will be coming. But we've got to be able to get the entire portfolio of MABs, not just the ones that are innovated in developing countries themselves.
Thank you, Burke. Thank you very much for that, Burke. No, we definitely do agree, and I think the speakers will just also echo some of those sentiments as they close. Peter, what was your question, please? Can we hear your question as well? Please come through. To all the speakers, we'll give you an opportunity to answer holistically and then to close up the session. So yeah, let's just wait. Peter, over to you.
Yeah, thank you very much. Maybe one of the questions we should be asking as well is that, is WHO overwhelmed? Because we have seen instances whether we talked about the time it takes for approval or authorization. We know what happened to Paxlovid, which was there for COVID, and it took too long, not only to about authorization, but even the guideline for administration took too long. So is it time that we think of other authorization bodies like the EMA, the CDC, to be given same portfolio so that we don't take too long for science to work on products that cannot be accessible because of the long chain of authorization? That's my thinking.
Thank you very much, my leader, Peter. I just wanted to read out Louis de Gama, who we are collaborating with through the community's delegations questions and assertions, because he cannot speak, he's in a public space. And he says that, you cannot demand something you do not know exists. So treatment literacy of the critical import of MABs is essential. Creating a 101 for monoclonal antibodies that is community friendly is essential. And can we commit to producing something like that, at least as a start off, because we need to build the capacity of community volunteers in order for them to then, I think, as we said, do the advocacy. And I just wanted to mention, thank you very much for that, Louis. And then in the chat Q&A session, we had Yolanda Augustine, who actually shared, kind of like a study, a proof of concept, where a group has been working on plant molecular farming for monoclonal antibodies for cancer. So clearly there are cases out there, right? So it's not like we're working from nowhere, number one. Number two, it's also the fact that these things are being used, or these MABs therapeutics are already being used in many high-income countries. Like if 80% of them are there and they're being used for a wide variety of communicable and non-communicable diseases, then it doesn't make sense why we don't have them. That is the question that I think Carmen will answer, but is also asserted to, which is we need to unlock this whole MABs thing, and that is what we want to do. So I don't think this will be our only L&D session on MABs, but this is the beginning of a conversation. I think there is a lot of resources out there, and I think what we can do as the NGO delegation is commit to sharing those. And then of course Sharon Ann is doing a lot of this work with O'Neill, partnering with the IAVI, partnering with Wellcome Trust and other colleagues, as well as Unitate. So we really do have a way forward, and if we work with colleagues such as, of course, TNW+, we will make sure that we've got the regulations in place, and of course, as Brooke and as Achal mentions, if we can just have a test case of government use around MABs, we'll have shown that we can do this. So clearly there are pathways. Clearly we have strategies that we can put together. It's how we pull together something that is coordinated, that makes sense, and that actually is sellable, because the money is out there. I don't think it's the money. It's about how we sell the topic matter, especially when 80% of people have it and there are continents which have less than 1%. As we close the session, I'd like to give my speakers just 1 minute each to round off and leave us with their parting messages and just like some sort of next steps message about what they think we can do with this. Sharon Ann will be the last to close out the session and to close us out. So let me hand over to you, Carmen, and then we'll hand over to, if, I don't know if you're still here. Is Dr. Mutu still here? Oh, he's
No,
still
she
here?
had to get off.
Okay, she had to get off. All right, so we'll have it as we have we'll have Carmen, we'll have Achal, and then we'll have Sharon Ann close us off. Thank you.
Thank you, Lynette. So you anyway said what I wanted to really close with, which is that our aim is to unlock this conversation. And I think that is extremely important. And first of all, because there is not much time, I saw the pipeline, and although it's not yet here, I mean, there is for some, like respiratory syncytial virus, which 90% of the mortality happens in low middle income countries, but others are in the pipeline, but the pipeline is not so distant. There are like maybe 2 years until the first one coming to. So we don't have much time, and that's the issue. We want to do everything at the same time because we know it's the only way to do things, to anticipate access. And at the same time, as we are trying to see about bioavailability for a business, for generics or for expanded production, we know that IP and all those other barriers must be addressed very early on. Otherwise, it will then take 2 more years, 3 more years after the products come out of the pipeline. So this is the call to help for help, because we really need to be strategic and think how we can put a roadmap where all those things can happen at the same time, the risking where there is a need to the risk those stakeholders like investors and developers or manufacturers for them to move forward. And also, a last comment is on communities. In the call, I didn't describe with detail, but you can see in the web or in my slides, one of the 4 outputs or work that we asked for in the call for proposals was engagement with communities. So this has been absolutely clear for us in this call, in this work, that engagement early on of communities at this stage is absolutely key. So very much supporting some of the ideas that have been put in the chat about how to start now with communities work, and really thanking the opportunity to discuss with you today. It's a very complex issue and we will need to continue the conversation. Thank you very much. Over. And thank you for the support to Unit call. Thank you very much.
Achal?
Thank you, Lynette. Lynette, firstly, thank you for so expertly guiding this conversation through. I really appreciate it. And thanks to Unit. I think that, to borrow Sharon Ann's language, actually, I think it's an ecosystemic problem. You have so few biosimilars. They're so expensive. Even when biosimilars come to market, their cost relative to the cost comparison between a generic small molecule and an originator small molecule, it's not the same. They're much more expensive relatively compared to generic small molecule drugs, right? That leads people to say, oh, they're too expensive. There's so few of them, they can't possibly be of value in poor countries. And so then that affects the entire research pipeline and this, vast, again, ecosystemic problem, right? And what I want to say is the one way in which it is similar to HIV, even though I spent a lot of time explaining why some of these problems aren't, is that the threat is diffused here, right? There are almost no autonomous ways that are being enabled for countries to make MABs if they wish, right? And that's why addressing the IP barriers and addressing and easing the regulatory burden will constitute the necessary threat. Brooke briefly mentioned the idea of transferring know-how or having cell banks where, what's called the original cell line, which is what the basis of a biologic drug, for instance, should be deposited so that it can be reproduced. Now, of course, technology transfer, transfer of know-how or even the depositing of cell lines will slightly aid the development of a biosimilar. And we've spoken extensively to manufacturers of biologics. This is one of those situations where transfer of technology is a sort of juju. How do you translate? Like a magic. It's part of the magic that is picked up around these monoclonal antibodies and biologic drugs, right? Without this, you're not going to be able to do that. It's not true. So we have an autonomous pathway. Yes, there are ways to make it easier if you licensed a biologic, perhaps, right? And provided all of these things. But it's by no means impossible, and it's a very small difference to recreate these things, right? So we need to set up that pathway, because even if we want them finally to be licensed officially and formally in the way that generic drugs are now, we still need to have a reason to give them to do that, like we did with generic drugs, especially for HIV so successful. Thanks.
Just 3 comments, because I already talked a lot. So obviously we have a lot of work ahead of us. Growing literacy, as people have said, growing demand, as people have said, reducing the markup for existing MABs. It doesn't need to be that high. And if my colleague from MSF, Julian Potet, was here, he would make that case, as he has it every time I've been in the room with him. And of course, facilitating and spurring biosimilar competition to bring prices down across the board. Thirdly, we need to bring about a whole shift in terms of, so that we can say eventually that those are the MABs from the before time, and that this is the after time. And that is, again, the manufacturing strategies and the different optimization strategies. So that also there's a coalition of the willing among public and private sectors, the developers and manufacturers, but then that also starts to become the new norm. Part of the reason you don't have MNCs investing in optimization right now, they've already built the big factories, the big machines, the chows, the reliance on the status quo. And listen, another lesson from HIV, when AZT was first developed and sold, it was set at such a high, incredible dose. And it wasn't until activists banged on their door in terms of this could be lower for the sake of safety and for the sake of affordability. Again, there is no incentive for the MNCs to reduce the cost of goods. So we need to develop an alternative and provoke that shift in the paradigm and provide a new norm. Thank you so much.
Sharon was closing us off. So thank you very much for joining us on this wonderful, and really critical webinar. Thank you, Carmen. Thank you, Achal. Thank you, all of you, the speakers, who joined and who spoke at the session. We will have a follow-up session on the same topic. We still owe you another follow-up L&D session on local manufacturing. So I think we've got a lot of work ahead of us in keeping with the trends in terms of where we're going, because all of these issues are interlinked, right? It's just how do you coordinate them and how do we support partners such as, for instance, IAVI, Unitaid, Global Fund, whoever it is, Wellcome Trust, whoever is working, and of course the TNW gorgeous guys and the IBA, the Access IBSA to come together and basically get into this so that we can find a way to make monoclonal antibodies available. Like right now, as Carmen said, time is running out. We literally have the product, the pipeline here. So we will do our best, I think, as the NGO delegation, as well as the communities, to keep the information alive. And we will basically be bringing all of you back to a 3-week training session on MAB. So we'll do MAB 101, MAB 102, MAB 103, and then we'll be ready to advocate. We did it with ARVs. We had to learn ARVs till 10 o'clock in the evening. We can do this again. So thank you very much all, and have a lovely rest of your morning, afternoon, and evening. And thank you for the speakers, as well as expert panel and partners who have made this session possible.